Thank you, everyone. It's at the top of the hour, so I'm going to suggest we reconvene our meeting and get started. First, we will have Dr. Rituparna Das from Moderna presenting on the Moderna COVID-19 vaccine mRNA-1273, safety, immunogenicity, and efficacy in children ages 6 months through 5 years of age. Dr. Das, the floor is yours.
Great, thank you so much. Good afternoon. My name is Rita Das, and I'm the Vice President of COVID-19.
Do you mind just swapping it? We can see.
Oh, sorry. Thank you.
You're totally fine.
All right. Is that better?
Yes, perfect. Thank you.
Great. So we're here today to discuss the use of mRNA-1273 as a 2-dose primary series for the prevention of COVID-19 caused by SARS-CoV-2 in young children 2 to 5 years and infants and toddlers 6 to 23 months of age, where, as Dr. Fleming-Dutra just showed, the greatest increase in the burden of hospitalization with the Omicron surge was seen. The proposed 2 doses at the 25 microgram per dose level are to be administered 1 month apart. Our data in these age groups comes from Study 204, where more than 6,500 infants, toddlers, and young children were enrolled, and more than 5,000 received at least one 25 microgram dose of mRNA-1273. Overall, this represents a substantial safety database for these groups. The median safety follow-up times in both age groups are more than 2 months. In the early cohorts, which is the dose-finding part of the study, we have children with 7 to 8 months of median safety follow-up. Study 204 was conducted in 2 parts for each of the age strata. Part 1 was an open-label dose-escalation study and was conducted first to select a dose level for further testing in Part 2, which was the placebo-controlled part of the trial. Both 25 and 50 micrograms were studied in the 2 to 5-year-old children. The 25 microgram dose was chosen for these older children, so the 50 microgram was not investigated in the younger children. Overall, the 25 microgram dose was chosen for both age groups because it showed an acceptable tolerability profile and demonstrated a high likelihood of meeting the pre-specified immunogenicity success criteria for the study. After Part 1 was completed, a DSMB meeting occurred to ensure the committee's concurrence with the selected dose. Part 2 was designed to randomize the children in a 3-to-1 ratio to receive either mRNA-1273 or saline placebo. The children will be followed for 12 months after receiving their last dose of vaccine. The data we will present today focus on the randomized phase of the study. Overall, the demographics were well balanced between the vaccine and placebo in both age groups. The mean age in the youngest group was approximately 16 months, and it was 3 years in the older age group. Gender, race, and ethnicity also were well balanced. On to the safety from the study. The specific safety objectives included solicited local and systemic adverse reactions collected for 7 days post-vaccination. Unsolicited adverse events were captured for 28 days after each vaccination, and serious adverse events, medically attended adverse events, and adverse events of special interest, including myocarditis, pericarditis, and MIS-C, were followed throughout the entire study. Starting now with the local reactions in children 2 to 5 years of age. In this figure, mRNA-1273 is shown in blue, and the placebo is shown in gray. Pain was the most common event, with similar rates and severity following dose 1 and dose 2. Most local AEs, including pain, were grade 1 or grade 2, with few grade 3 reactions. The median duration of local adverse reactions in this age group was 2 to 3 days. Looking at the infants and toddlers 6 to 23 months old, pain again was the most common local adverse reaction, although reports of pain in this youngest group were more similar to placebo, both post-dose 1 and post-dose 2, compared to the older age group. Systemic adverse reactions were evaluated according to age. Young children's events were fever, headache, fatigue, myalgia, arthralgia, nausea, vomiting, and chills. For infants and toddlers, the events were fever, irritability and crying, sleepiness, and loss of appetite. Headache and fatigue were the most common systemic adverse reactions in the children's 37 months to 5 years. Among the vaccine recipients, systemic adverse reactions were more frequent post-dose 2 compared to post-dose 1, although this difference was less pronounced than we've seen in older age groups. Duration was 2 to 3 days, very consistent with other age groups. In this slide, we're showing the systemic adverse reactions collected for infants and toddlers. On the top are the 24 to 36 month group, and on the bottom are the 6 to 23 month group. Here, reporting rates of systemic adverse reactions were similar between dose 1 and dose 2. Also, these systemic events were reported at similar rates among vaccine and placebo recipients. I will discuss fever separately since this is a particularly important event in the assessment of pediatric vaccines. This slide shows fevers by increment among children 6 months through 5 years. Overall, fever after any dose occurred in about a quarter of the children. The distribution of temperatures was similar between the two age groups. Fevers of greater than 40 degrees Celsius were rare. In this slide, we have the maximum temperatures post-dose 1 and post-dose 2 according to temperature ranges shown on the top. On the top are fevers in the children 2 to 5 years. We see the fevers occurred more frequently following the second dose. It's important to note that most reports of fever were less than 39 degrees Celsius. On the bottom are fevers in the infants and toddlers. Again, fevers are reported more commonly post-dose 2. The rates of fever greater than 39 degrees in this youngest group are similar to placebo. This next figure shows fever by day after the second dose in children 2 to 5 years of age. Most events of fever occurred within 2 days following vaccination. Beyond day 2, we see that fever rates in children receiving mRNA-1273 are similar to placebo. The median duration of fever in this age group was 1 day. It's important to note that this study was conducted during the winter months, when respiratory tract infections are prevalent. This is evident by the relatively high rates of fever that we observed in the placebo group. We see a similar pattern in infants and toddlers, with the peak of fever occurring on days 1 and 2 after vaccination. On subsequent days, fevers look similar across the vaccine and placebo groups. The higher background rate of fever is even more prominent in the infants and toddlers. There were 15 children with fever greater than 40 degrees Celsius in the mRNA group and 3 in the placebo group. The peak temperature of greater than 40 degrees Celsius had a duration of less than 1 day. Of the 15 vaccine recipients, 6 of the children also had symptoms of concurrent viral infection. There was one febrile seizure which occurred proximal to vaccination and was considered related by the investigator. You'll recall that febrile seizures can occur actually in up to 5% of young children. The child in this study also had a maculopapular rash onset 2 days after the seizure and went on to have a subsequent seizure associated with another fever approximately 6 weeks later. The child has remained in the study and received dose 2 of the vaccine without event. Next, I'll discuss unsolicited adverse events. Presented here are the events reported up to 28 days after any injection in children 2 to 5 years. The incidence of unsolicited AEs was similar among vaccine and placebo recipients. There were no SAEs considered to be related by the investigator. Incidence of medically attended AEs were similar, and there were no AEs which led to discontinuation of the vaccine or from the study. After the data cut, one event of urticaria was reported on day 1 post-vaccination that did lead to discontinuation. There were no events of death or adverse events of MIS-C or myocarditis. Among the infants and the toddlers, the incidence of unsolicited AEs overall and the MAAEs were similar among vaccine and placebo recipients. There was one SAE within 28 days, which was considered related to vaccination, which I already described in the fever discussion. Since the data cut of our submissions, which was in late February, we pulled the SAEs from the live database in early May to provide further reassurance of the long-term safety of mRNA-1273. The updated analysis did not identify any new safety signals, and there were no SAEs which were considered by the investigator to be related to vaccination. Next, we'll turn our attention to the immunogenicity data. Vaccine effectiveness was successfully inferred by meeting the predefined co-primary immunogenicity objectives, which were agreed with the FDA. These included geometric mean concentration and the difference in seroresponse. In each age group, immune responses were compared to the 18 to 25-year-old immunogenicity subset from the 301 study, where primary efficacy of the vaccine against any and severe COVID-19 disease was demonstrated. In the middle of this slide is the immunogenicity from children 2 to 5 years. The two co-primary immunogenicity objectives were met for these children. The ratio compared to young adults was 1.01 with a lower bound of 0.88. The seroresponse rates were close to 100% with a difference of minus 0.4% with a lower bound of minus 2.7%. Now looking at the infants and toddlers 6 to 23 months of age on the left column in the lightest blue, we see again that both co-primary immunogenicity endpoints were also met. The ratio compared to young adults was 1.28 with a lower bound of 1.12. In addition, the seroresponse rate was 100%. The group difference of 0.7% was seen with a lower bound of minus 1.0%. The immune response to mRNA-1273 has been remarkably consistent across age cohorts despite administering lower doses to young children. This slide depicts the immune responses ranging from young adults, 18 to 25, through the youngest children in our pediatric program. Across these age groups, the ratio ranges from 1.01 through 1.28, successfully meeting all primary immunogenicity hypotheses in all age groups. Additionally, support for vaccine effectiveness was provided through assessments of efficacy in our study, which were secondary, which demonstrated that in all age groups, we had comparable effectiveness to adults when the variants were matched. Although we did not observe any severe cases of COVID-19 in the children at the time of the data cutoff, this consistency leads us to believe that protection against severe disease will be comparable to adults, which will be evaluated in post-authorization effectiveness studies. So to expand upon this a little, we can see here how the studies were conducted as the SARS-CoV-2 pandemic in the U.S. was changing. This is important when interpreting the efficacy results in our two youngest age cohorts. The enrollment and efficacy follow-up in the youngest children was conducted when the Omicron variant was predominant. And the impact of the Omicron daily incidence is apparent from the curve that's shown in red. From December 2021 through March 2022, the U.S. incidence of SARS-CoV-2 infections rose from fewer than 200,000 per day to a peak of 1.4 million cases per day, which is when these youngest cohorts were followed. now to the efficacy results. While the study was not powered for assessment of efficacy, we were able to get good efficacy estimates given the high rates of community SARS-CoV-2 transmission when the randomized cohorts were conducted. Two case definitions for COVID-19 were used. First, the CDC case definition, which required at least one symptom, and then the Study 301 case definition, which required two systemic symptoms or one respiratory symptom. The CDC case definition was considered primary since children tended to have less severe symptoms of COVID-19 than adults. Of note, both definitions required a nasal swab positive by RT-PCR for SARS-CoV-2. We took a comprehensive approach to capturing cases. Efficacy estimates in the young children are based on 180 cases captured over 71 days post-dose 2. Beginning with the children 2 to 5, there was a lower incidence of COVID-19 by both definitions in children who received vaccine compared to those who received placebo. Statistically significant vaccine efficacy of 36.8% was observed using the CDC definition. When we get to the P301 case definition, we see how the number of cases is reduced, but efficacy of 46.4% is observed. This is also demonstrated by the cumulative incidence curve for COVID-19 by the CDC case definition, and this is in the MITT1 set. The mRNA group is depicted in red and the placebo is in blue. The separation of the lines is observed shortly after dose 2 of the vaccine, which is evidence of the early onset of protection. Next, looking at efficacy among infants and toddlers 6 to 23 months. Efficacy estimates here are made on 85 cases captured over 71 days post-dose 2. We see again statistically significant efficacy of 50.6% when the CDC definition is used. When we see the P301 definition, the point estimate is directionally similar, and the confidence intervals are wider due to the drop in the number of cases. To that end, at the start of the Omicron wave, we noticed that parents were reluctant to bring the youngest children into the site for illness visits. Instead, they were calling in results of positive home antigen tests. So since we also captured the results from home antigen tests, we did a sensitivity analysis defining COVID-19 either by positive PCR or home antigen tests. With the increased number of cases captured, the confidence intervals narrow, and the point estimates for efficacy are 53.5% and 43.7%, with the confidence intervals excluding 0. This is also demonstrated in the cumulative incidence curve for COVID-19 by the CDC case definition in the younger age groups as well. The mRNA group is depicted in red and the placebo is in blue. The separation of the lines again is observed shortly after dose 2, evidence of the early onset of protection. For context of the efficacy, we'll now look at real-world effectiveness in adults during the Omicron surge to help interpret the vaccine efficacy from our pediatric program. Presented on the left are the real-world effectiveness data against Omicron after a 2-dose primary series among adults. These data are from our collaboration study with the Kaiser Permanente health system. Vaccine effectiveness of mRNA-1273 against COVID-19 was 44% when the Omicron variant was predominant. On the right are the estimates of efficacy from Study 304 in infants and young children. Efficacy of mRNA-1273 was consistent with effectiveness observed in adults. While vaccine effectiveness against any infection was lower during Omicron, we continue to see that the benefits of mRNA-1273 continued against hospitalization. 2 doses of mRNA-1273 was shown to be 84% effective against hospitalization during the Omicron period. This is important because we would expect the same level of protection in children given the consistency of the immune response and the efficacy with adults. Study 204 is ongoing and safety follow-up will continue for all participants. Children will all be offered a booster dose after their second dose, and they will be boosted either with mRNA-1273 or our bivalent Omicron-containing vaccine. So in summary, mRNA-1273 was well tolerated. Local and systemic reactions were seen less frequently in these youngest age groups. Solicited adverse reactions were mostly grade 1 or 2 and slightly more common after dose 2. Fever was reported in about a quarter of children and most commonly seen on days 1 or day 2 after each dose and resolved within 1 day. No deaths, myocarditis, pericarditis, or MIS-C were reported among vaccine recipients. There was one related SAE of fever, febrile seizure within 28 days of any vaccination. The primary immunogenicity objectives were met. 2 doses of mRNA-1273 were shown to be immunogenic. GMCs and seroresponse rates were non-inferior to young adults. Vaccine efficacy can therefore be successfully inferred based on immunogenicity. In both age groups, direct efficacy against COVID-19 was observed during the Omicron period, again consistent with effectiveness observed in adults. Based on the information we've shown here today, we've demonstrated that the benefit-risk profile for mRNA-1273 is strongly favorable in children 6 months through 5 years of age. I'd like to take a moment to acknowledge all our study investigators, site personnel, our colleagues at BARDA and the COVI-PN and NIH, and really most importantly, the infants, toddlers, and children who participated in the trials and their families. Without you all, our pediatric program would not have been possible. Thank you very much, and I'll be happy to take any questions.
Yes, it does. This presentation is now open for questions, and I see Dr. Paling has her hand raised. Please go ahead.
Thank you, Dr. Das, for a very clear and informative presentation. I've got 2 questions. The first is, could you clarify what the placebo is? And then the second is, we saw that some children probably had COVID-19 in the past. And do you have any data about those that were seropositive before being vaccinated? Thank you.
Yes, absolutely. So the placebo was saline. And then let me pull up a slide. We had about 6 to 8% of children with baseline either seropositivity or a day 1 SARS-CoV-2 positive from a nasal swab. And so we had immunogenicity data from those children as well. And I can share them now. Okay, here we go. Okay, and you see here, so make this a little bit larger. Yeah, you see here that the, for the seronegative, I'll call them seronegative, we had for the 2 to 5 years, the GMC was 1425. For the seropositive children, it was over 6,000. And for the 6 to 23 month olds, for the seronegative children, their GMC was about 1,700. And for the seropositive children, it was much higher.
Thank you. I see Dr. Sanchez.
Yes, thank you for that presentation. So getting back to Dr. Paley's point, the data that you have presented, did it exclude the ones who were seropositive?
No. We included them for, we did not, the seropositivity was checked at trial entry, but it was not an entry criteria into the trial. So
the data you presented earlier, it included both.
Yes, so the safety data I presented did include both. The immunogenicity data I presented only includes per protocol immunogenicity, which is only the seronegative. And then I just showed the seropositive immunogenicity data as well. I can put that one back up. And
that's okay. I just wanted to clarify.
Okay.
But actually, I had another question. In the 6 to 23 months, where was the vaccine administered?
So it was left to the to our investigators' discretion to be administered in the arm or in the thigh.
And so we only saw data about axillary swelling. If it was administered in the thigh, was there any inguinal adenopathy seen?
Yeah, for those youngest, actually, we allowed thigh administration, I think, through both age groups. Oh. And our swelling was measurement was proximal to the site. So the question that was given to the parents and to the investigators was axillary or groin swelling.
So then you did see some inguinal swelling or groin swelling?
Yes, we did.
Thank you.
Thank you. Ms. Bata.
Thank you. And thank you, Dr. Das, for this nice summary. One of the questions I had when I was looking at the systemic reactions, particularly related to fever, I was seeing some really higher fevers on day 4 and day 5, and then you were talking about children who were PCR positive with the first dose. What kind of inclusion criteria did you have related to respiratory infections?
So we, yes, so I can put up the fever slide again. Apologies, let me put this up. Okay, so this is a 2 to 5 year old fever slide. And as you can see here, the top is the vaccine, the bottom is the saline placebo, and we see most of the fever being on day 1 or day 2. And then the hash is the greater than 39 here. So beyond day 1 or day 2, there were some there were some in the hash greater than 39, but it was fairly rare after day 1 or day 2. For the inclusion-exclusion criteria for the study, they did not. The children were not supposed to be acutely febrile when they were in the study, but if they had like cold symptoms or something, they were allowed to be in the study, and they just could not have been SARS-CoV-2 positive or like known SARS-CoV-2 positive in the past 14 days for study entry.
Thank you.
Thank you. Dr. Paley?
So thank you again. I have 2 questions. One is, we understand that the goal was to vaccinate within 4 weeks. And could you share the extent, what the range was, and or what percentage of them would have been vaccinated more than 8 weeks apart? And the second is, can you please clarify what percentage of children had underlying conditions? Thank you.
Okay, yes. So our investigators and our study parents were incredibly compliant. I think it was over 90% vaccinated right within 4 weeks. There wasn't enough variability to really warrant analyses of like significantly longer ranges. And then, Dr. Paley, you had a second question, and I'm sorry, I forget.
What was the underlying conditions that were included, and some examples?
So the underlying medical conditions, I do have a slide on that one. Okay, maybe we didn't pull this slide, but so we had, obesity of about like 15 to 20%. We had asthma or chronic lung disease for about 5% or so. It was a fairly representative population. Those seasonal allergies. This was a fairly representative population of what a general pediatrics population would be. We did exclude more severe immunosuppressing conditions, and well-controlled HIV was allowed, but I think we only had 2 or 3 children with HIV in the study.
Thank you. Dr. Daly?
Yeah, thanks so much. I'm going to continue along with this sort of line of questions, which is what proportion of the safety data set were seropositive at baseline? And then there's a follow-on question, which is, did the reactogenicity, particularly systemic reactogenicity, including fever, differ among those who were seropositive and seronegative at baseline?
So for both age groups, we had a baseline 6 to 8% seropositivity. And we've analyzed this carefully by baseline seropositivity for all of our age groups. There's a preview to our presentation next week. We did notice some increased systemic reactogenicity in the older children, so 6 to 11, and in the adolescents. But it was, they looked more like post-dose 2 after dose 1, and then their post-dose 2 was not any different. For these youngest children, and I do have that slide, for these youngest children, fever, I meant to share, but I didn't actually. Here. For these youngest children, it was really only fever that was different. So post-dose 1, the blue is the baseline seropositive, and the black is the baseline seronegative. We saw a bit more fever after dose 1. After dose 2, it evened out. For the younger, this is for 2 to 5, and then for the younger children, we saw kind of the same thing. There was a bit more fever, but the rest of the solicited adverse reactions were not different.
Great, thanks so much. And I have Dr. Lee, while I've got the floor, can I ask 2 follow-up questions?
Go ahead.
Great. So, Dr. Das, for fever, do you know if any of the children who experienced fever sought emergency department care or were hospitalized for that fever or had sort of workups for that fever?
No, there was the one child who had the temperature of 103, who had an ED visit for the fever, and then it kind of, it was an unwitnessed seizure, but it was assumed to be a seizure based on some decreased responsiveness. That child was taken to the ED, observed, and discharged. There was no other reports of ED visits or hospitalizations related to fever in the study.
Okay, great. Thanks so much. And then one last quick question, and then I'm done. That question is, I understand that you are studying boosters in this age group. When would data on safety and immunogenicity of boosters be available for this age group? Over.
Yes. So as as we've gotten the authorization by the protocol, the study unblinds now. And so we have a rollover study starting next week, where we will provide the Omicron containing booster to the under 6-year-olds in the study. And so that booster portion, it'll depend on how quickly they enroll. We think it'll be very quick, and we are hoping, so then we'll get to day 28, and then the time it takes us for testing. So we hope by early fall, we will be able to bring forward the booster immunogenicity and safety data for this age group.
And just to clarify, what would be the interval between dose 2 and the booster dose?
We've discussed this interval quite a bit. When we boosted adolescents and we boosted 6 to 12 in the study. And so when we started this, we started the boosting at 5 months, which is congruent with the adult boosting. Now, I think it's kind of hard to understand, is it waning immunity or is it Omicron that is kind of driving the need for boosters? And so, we have safety data from adults for boosting at a minimum of 3 months. And so we are going to boost 3 months or more after the primary series.
Thank you. Back to Dr. Sanchez.
Again, thank you again. You mentioned a couple times that some of the fever and adverse effects could be attributed to concurrent viral infections. And was that verified, or was that because, I mean, were you able to document some adenovirus or RSV or something else to back up that statement?
Yeah, so there were 6 of those cases where there were the concurrent symptoms, but this is based on the concurrent symptoms that are reported by the PI. I don't think we had like an alternate virologic call.
And one more question. Any of these these children, particularly the younger age group, were they preterm? Had they been born prematurely?
Yes, I believe we had 37 weeks and up was our gestational age criteria, but I'm going to confirm with our. Okay, yeah, they could be more preterm if they were over 1 year of age. And so we did not have any 37 weeks and up was considered term. And then they had to be, the preterm infants had to be at least 1 year to be in the study. So we did not directly have preterm infants in the youngest cohort.
And is there any plans to look at the extremely premature infants in terms of their dosing? I mean, we dose other vaccines on a chronologic age, but are there any plans to look at the 24 to 36 weekers at a chronologic age of 6 months?
No, that's a very good question. And as a pediatrician, that's also near and dear to my heart. So we are going to be starting a study in even younger infants that we're going to call Baby Code. And we're under discussions now for the greater allowance of prematurity in that study.
Great, thank you. Very much needed.
Absolutely.
Dr. Lair.
Thank you very much, Dr. Doss, for your presentation. Could you go to slide 5, please? I'm wondering how organization, pharmaceutical company decides on dosing. And you look like you did the dosing, compared 2 doses for toddlers or for young children, but not for infants and toddlers. I'm just wondering how you made that decision. Thank you.
Yes, so you're absolutely right. We did the 25 micrograms and the 50 microgram dose. We looked, and then we had the, and so when we saw that there was a bit more reactogenicity for the 50 micrograms, we did not pursue the 50 micrograms in the youngest children. But after we enrolled all the youngest children at the 25 microgram dose, we looked at the immunogenicity from part 1, and we saw that the immunogenicity actually looked like it would meet the pre-specified immunobridging criteria, but there was not necessarily a lot of room to go down. And the tolerability profile for the 25 micrograms in the 6 to 23 months actually looked quite good. And therefore, we chose not to look at an even lower dose in the 6 to 23 month old group.
Thank you.
Thank you, Dr. Brooks.
Yes, thank you, Dr. Doss, for the presentation. I have 2 questions. One is, you stated there were no myocarditis cases found. Did you give the parents any specific symptoms to monitor, as myocarditis would present differently in especially the 6 month to 23 months and even the 2 to 5 year olds? So how did you determine there was no myocarditis?
Yeah, no, that's a very good question. We talked to, so when the myocarditis signal was identified, we updated all of our informed consents across our whole adult and pediatric programs. We did a training with our investigators, And we actually started to ask about any myocarditis symptoms in our routine unsolicited AE phone calls. And so we developed a script and we asked about all of the CDC components of the definition with the parents. And so, for the adolescents and the 6 to 12 and the 2 to 5, I think those symptoms would have been more directly reported, as you said. For the youngest children, I think it did get more challenging. And this is, we did educate the investigators that, symptoms of myocarditis present differently in this youngest age group and what those symptoms were. It's tough, I think, in general for myocarditis in the youngest age group because the symptoms are fairly nonspecific. And so a lot of it does overlap with the reactogenicity symptoms that we were soliciting for. So we told the parents that if something, or the investigators, that if something is lasting particularly long or looks unusual, please bring the children in for evaluations. For all around, if there was any suspicion for myocarditis, we asked the PIs to bring the children in for evaluation.
All right, thank you. And my second question is related to immunogenicity. Since you had this serum, did you do any T-cell studies to see if there was any cellular immunity that you could measure in terms of a positive response?
Yeah, so we have the blood collected for actually all of the age groups. The cohorts are small because of the blood volumes that are required, but we have analysis plans, and hopefully we can bring the T-cell analysis forward that can, kind of ensure that directionally it's similar to what has been seen in adults. We hope in the next quarter to bring that forward.
All right, thank you.
Thank you, Dr. Daly.
Yeah, Dr. Das, I want to follow up on a comment that you just made about dose in the 6 to 23-month-olds. I mean, acknowledging that finding the dose it's a balance between immunogenicity and reactogenicity, and I recognize that. And also there's immunization delivery implications. But you said you didn't think there was room to go down on the dose. And if you could just clarify if you meant in terms of meeting immunogenicity criteria or some other reason. Over.
Yeah, it's solely for meeting the immunogenicity criteria. As you know, we were asked to make sure, well, our study had that the GMR ratio had to be 0.8. And in our discussions with the FDA, the preference was that if lower doses than the adult dose, than the 100 microgram dose were being investigated, that GMR ratio should not be lower than 1. And so we were not confident about meeting that GMR of 1 if we went with a lower dose. And that's why we went with the 25 micrograms. That said, when we're doing our Baby Cove study, we will be doing more kind of down dosing of the immunogenicity.
Thank you. Dr. Hoag?
Thank you, Dr. Das, for the presentation. We appreciate it. I'm wondering if you could just remind us, I'm sure many people have questions about, this is the age group where we're giving lots of routine immunizations from this age. Most of our routine immunizations, in fact, fall in the age group that you're presenting data on. Can you remind us about whether the children in this study were also during this time frame receiving their routine and recommended vaccinations, and any comment on concomitant administration of the vaccine, of your vaccine with those routine immunizations? Over.
Yeah, no, that is a very good point. We did not allow concomitant immunization for this first, for kind of this first pass study. We did ask the investigators to offset flu vaccine by 2 weeks and offset other routinely administered vaccine by 4 weeks. And so we do not have concomitant administration data yet. Fully take the point that that is an important piece, and our future studies will look at concomitant administration, both in terms of immunogenicity and reactogenicity.
Thank you. Dr. Paling.
Thank you. I've got two questions. One is, could you share what you learned about lymphadenitis? We do know that these vaccines are reactive and lymphadenitis is relatively common. And then the second is, on your upper age, children 5, were there any that were enrolled and by the time they got to the second dose were eligible for the next dose? Thank you.
Yeah, so I will take your second question first. We, so we did not adjust the dose. I know the age cutoff can kind of be arbitrary, but our investigators followed the rule that they got whatever dose they started with. So if they started with 25 micrograms and they turned 6, they would still get a 25 microgram dose. So they would stay within the cohort. In terms of the lymphadenopathy, we did see lymphadenopathy, the incidence of lymphadenopathy in children. I believe it was a bit lower than, it was a bit lower than, I shouldn't make the statement about the comparison with adults. Actually, the number is escaping me. But for the 2 to 5-year-old age group, we had that it was about post-dose 2, it was 9% in the vaccinees, but it was all grade 1 or grade 2. There was no grade 3 or grade 4 lymphadenopathy. For the youngest children, it was also, again, about 9% post-dose 2, and it was all grade 1 or grade 2.
And could I follow? Could you
Sure.
clarify the time frame of resolution?
No, I just have the data in front of me for the within 7 days. I know we did the analyses for the resolution. I don't have them at my fingertips, but I will follow up with that question.
Thank you. Dr. Brooks, you have your hands raised. I just want to make sure. Did you have another question? Okay, I'll assume not. I did want to follow up on a point, and now I can't remember which colleague of mine made the point, which is this question about concomitant vaccination. It has to do with whether or not, and perhaps it's a question for Moderna/FDA, whether or not the labeling will include information about the risk of febrile seizures, which can happen with any infection or with vaccines, and what that might look like. And then just thinking about our clinical considerations team and also the AAP and PIDs, whether or not there's any comments about at least providing information to families about the risk of febrile seizure generally and then specifically.
I can start by what is in our approved fact sheet. So in our approved fact sheet, we do have the case detailed out, the case of the febrile seizure that occurred after the first dose. I'll leave it to others to talk about the rest of the questions.
Yeah, I'll defer. This is Dr. Oliver. I'll defer to Dr. Fink if he wants to comment on the EUA specifically, but I can say we have a presentation tomorrow on clinical considerations, and febrile seizures will be mentioned there, but I can save the discussion and the exact language for tomorrow.
Okay, that's absolutely fine. We can discuss it tomorrow. Thank you. Are there any other questions for Dr. Das? I don't see any additional hands raised. So as planned, we will take a break until the top of the hour, so 1 p.m. Eastern Standard, 10 a.m. Pacific time, and we will return to hear Dr. Gruber from Kaiser. I want to thank Dr. Das for her presentation and for patiently answering all of our questions, and we'll look forward to talking to everyone very soon. Thank you.
Thank you so much.